Yeah, so thank you very much. It's a tremendous pleasure to be here. A very interesting meeting, interesting crowd. I had a very nice dinner also last night, so the discussions are really inspiring. So I normally never think about bringing synthetic cells to space, but I guess now I am. And so later today, there's going to be a session with the title Emigration of Life from Earth. And I guess the talk that I'm going to give right now relates mostly to that session. And it's, I guess, to entertain the idea, the thought that if we ever decide or manage to bring life in a controlled way away from Earth, you know, depending on the outcome of the discussion, whether we should, we could think about other forms of life than the natural one that we know on Earth, and maybe even think about artificial cells or synthetic cells. And the advantage could be that these could be very simple, controllable, cell-like, life-like entity cells that you could maybe use for production of materials wherever you might need that. And actually for quite a lot of things that you might also want to use these synthetic cells here on Earth. So as was already mentioned, it's bringing synthetic cells to space, big question mark. And the big question mark is also because we do not have synthetic cells. So the real title actually is building synthetic cells, big question mark, because we're still far from that, even though there's a lot of enthusiasm and effort and sort of momentum, both here in the United States and in Europe and Asia to really make this happen in some form in the next, whatever, Drew thinks 5 years, we think 15 years, so it's a different level of optimism at two sides of the ocean. So what I will do is talk about, on the one hand, some of my own work, my own sort of inspiration or you know, the kind of thing that we usually do in the lab that led me also to being involved in this project, but also talk about the community building that we are doing around this topic in Europe and in the Netherlands in particular. So I will show you, we're running since recently a national program where we put different people together to really try to make this work, and at the same time building a community between scientists and also industries in Europe to do this. And I know the same thing is happening in parallel also in the US, and I will not talk about it, but Kate maybe will refer to it later. And this is sort of going on in parallel, and we talk to each other a lot, which makes it even better. So that's going to be the mix of this talk. So I'm in the Technical University of Delft in the Netherlands. And I don't know, anybody ever been there? Good, you're welcome. We could have one of these maybe there. We have a very strong faculty of aerospace engineering and also planetary research that I know very little about, but you probably know some of my colleagues there. So, but let me start by sort of what fascinates me and why I'm interested in, or have been interested in the idea of rebuilding or reconstituting, as we sometimes say, cells from scratch. So I always show this movie, no matter what talk I give, mostly to students, and I always ask who has seen it before, but here maybe I'll ask who has not seen this movie before. Okay, so there's still a few. So this is just a small piece of a movie that's called Inner Life of the Cell, and it's showing an artist's impression, obviously, of a molecular motor. It's a nanoscale machine. It's called kinesin, and it's what it's doing, it's bringing a small vesicle packed with proteins, presumably, or other materials, through the cell. So it's a transport mechanism, and it uses these rails, microtubules, to transport them. And this is a single molecule, actually. It's a dimer that uses ATP to generate, to produce work. So it's generating forces, and it's transporting things through the cells. So, and as a biophysics community, I was trained as a theoretical physicist, but then very quickly moved to experimental biophysics. I can still put it on my CV, but that's it. And, but as a biophysics community for the last 25 years, I think we've been very successful in figuring out the details of how these molecular machines work. So with the help of nanotechnology, super resolution microscopy, structural biology, biochemistry, all these different approaches combined, we've learned how these individual, at least many of them, work. This one makes steps of 8 nanometers. It burns 1 molecule of ATP every time it makes a step. I believe you've seen the ATP synthase machinery yesterday. I missed it, but it's another really beautiful example of how much we've come to learn about the working of these individual molecules. And that's great. But of course, what is really interesting is how all these molecules together manage to be organized in time and space within a cell, communicate with each other to make this possible. So this is now a movie from a Japanese lab from Professor Niki, showing a simple unicellular organism. It's not a bacterium. It's, as you might imagine, because you can see a nucleus there filled with green DNA. It's a fission yeast cell. It's about 10 micrometers long. And this cell is sort of exemplifying what all life can do, whether it's simple or complex cells. And this is to somehow organize inside the container that defines the cell all these individual molecules in such a way that function arises. And there's a lot cells can do, but at a minimum, what all cells can do is grow and divide and transmit information, in this case in the form of DNA. So what we are really transitioning to also as a biophysics, biochemistry community is to go to much higher levels of complexity. So what also drives me is really understanding how these molecules that we know quite well now on the nanoscale work together to make this possible, this growing and dividing. And the question we ask, one of the questions we ask you, what does it minimally take? So what is the essential set of players, molecular players, that you have to put together to make something like this possible? And more importantly, of course, how would you have to put these together? And to come to some kind of what we call bottom-up minimal life. Now, there's another way of getting to minimal life, and this is a picture actually now already 3 years old, and this field advances quickly, so I'm sure it's not the latest information I'm giving you in this picture, but it's from the Greg Venter Institute, where there's what I tend to call, and others also, a top-down approach to minimal cells. So this is derived from a real bacterial cell. It's an electron microscope picture of a cell-like system, or a minimal cell if you want, that was made by taking a natural cell that has over about 1,000 genes, and then systematically stripping away genes. And then at the end, at least this point in time, they were left with 473 individual genes, and those were coding for the complete set of molecules that makes this cell alive. So this cell can grow, divide, transmit information. So the number of components, individual different players, is apparently overseeable, I would say. It's not thousands, it's more like hundreds of molecules. So that gives us some hope that by combining this number of molecules, we could also, by an alternative approach, bottom up, get to a synthetic cell. So it's a complementary approach, and it's very important, of course, that we communicate between these two approaches all the time. So just to repeat this by now a bit, a quote that is getting quite old, but the idea is, okay, you can make minimal cells and find out that how many components you need, but it's still very difficult to find out what each of these components is doing and why and which set of components you actually need. So the other way, as I said, is to just follow this idea that if, being engineers also in Delft, we should, if we really want to be able to claim in the end that we understand how these cells work, also be able to build them from scratch using the individual components. And like I said, that's really at the heart of my motivation. Cells can do amazing things, even the most simple ones. For the moment, I would say we have no clue of how this arises from the interaction between components, and by building it, we may get a much deeper understanding, even if we fail to ever build this cell to begin with. Just the path towards getting these synthetic cells will teach us a lot about how current biology works. And in case I will forget later, so this is not about the origin of life. It's not about the question, how did life once emerge? It's really about how does life as we know it now, based on the molecular components, DNA, RNA, proteins, as we know it now, how does that work? And if, of course, if we develop a much better understanding of that, there's all kinds of opportunities. And I'm just showing some random pictures, sort of emphasizing that if we know much better how cells work, We can use this knowledge, of course, in all kinds of application areas, but also if we actually manage to build simple cells like this that we can control, you might use them for, as I said in the beginning, for fields where we now use regular biotechnology, so to make biofuels, to make materials, maybe use them as carriers for drug delivery opportunities. So there's a whole range of applications you might think of, including what we're talking about today. Use them maybe as factories to synthesize molecules that you might want to use elsewhere outside our own world. So that's what the program is about. I said I will talk about, towards the end, sort of introduce to you the national program that we started 2 years ago in the Netherlands. It's called BASIC, and then also just tell you a little bit about the story, what we're currently in the middle of, and what was also right after this, in 2017, also started this initiative to also get a European larger scale program on this topic, which would probably include a much broader set of questions, including the origin of life. So, and some people in this picture you may recognize in this room. Drew was there, and so it's been very nice to constantly communicate between these two communities. So I will use a little bit of time to just tell you about where I come from, what my group is working on. And so we've been actually interested in the biophysics, the biophysical properties of the skeleton of cells in the context of eukaryotic cells. So we're only now transitioning from trying to do this kind of approach from big complex eukaryotic cells to prokaryotic cells. So what I'm showing you now, it has been sort of the work of my lab over the years, is mostly inspired by how the skeleton in eukaryotic cells work. So people shown here on the bottom are currently in my group, and then I list a few other names who've been important for the pictures that I will be showing today. Okay, so this is a picture taken by Torsten Wittmann, just to remind you that every eukaryotic, but also prokaryotic cell actually is not just a bag of chemistry. It's also, of course, a lot of chemistry, but it has a very important mechanical component to it, the skeleton. And the skeleton of cells, as probably you all know, it's not a static structure like our own skeleton, but it's a network of polymers that are extremely dynamic, that constantly change individual lengths, network properties, and can therefore also support different functions that cells have. So, and if you zoom in on one of these wires that you were showing in the that was showing in the previous picture, this is again taken from the inner life of the cell, showing that each of these individual polars is an extremely dynamic system that constantly changing length just by self-assembly of protein subunits into this hollow structure, and then switching between periods of growth and shrinkage. And what is interesting to me and what we've been working on is how this growing and shrinking filaments can actually generate forces, literally by pushing and pulling, for example, on chromosomes, and how the dynamic properties together with the forces that are generated help cells to organize space. So it's important that biochemical reactions don't happen homogeneously everywhere in the cell. It's important that chromosomes find their correct position in the cell, and therefore you need some kind of an internal spatial organization. And I'm not here to say that the skeleton is the only thing responsible for that, but it's playing a big part for sure in eukaryotic cells, but also in prokaryotic cells, as we find out more and more. So we've been interested in how these dynamic properties and the force-generating properties contribute to, for example, essential processes like cell division. So that's what I'll focus on today, but we're interested also in how this system helps cells to polarize, to move as really multitasking mechanical network that, depending on how you control it, can do an amazing number of things, including one of the most essential things that any cell, but also a eukaryotic cell does, is to divide chromosomes during cell division. So these are complete textbook movies, pictures, that you, I'm sure, are familiar with, showing how the chromosome pairs during cell division need to be first lined up in the middle of the cell and then eventually pulled apart to create two new daughter cells. And it's a very, to my mind, physical process where literally forces pushing needs to happen to put these things first in the middle of the cell and eventually separate them. And there's, of course, the vast amount of work of cell biology, biochemistry over the last decades that has taught us a lot about the essential players in cell division, all the different stages that the cell goes through, how it's extremely precisely and tightly controlled by biochemical checkpoints. And what we've been contributing is this bottom-up approach to try to build minimal systems that allow us at least to understand the mechanical aspect of this. So I'm just showing you the type of experiments we've been doing, and over the years we've been trying to build up these very simple systems initially to reach more and more what I call functional complexity. So increasing the number of components that we put together, such that we do not only look at the biophysical properties of these filaments, but eventually also learn about how they self-organize to be biologically functional. So, for example, all the way on the left I show each time on the top a cell movie, often coming from collaborators because we don't work with cells ourselves, and then at the bottom, the property that you see in the cell that we can reconstitute in a very simple system. And sort of the most trivial thing that we can reconstitute you see on the bottom, and that is the fact that these proteins that form these cytoskeletal polymers just by themselves are able to form these tubes that also spontaneously show this dynamic property. So you see in magenta pieces of microtubules stuck to a surface, and then green wires that spontaneously grow and shrink, just like you see in the cell. So you do not need anything else besides the protein and an energy source to make this dynamic property happen. Nothing else from the cell is needed for that. You also don't need anything else for these filaments to be able to generate forces. So on the top, you now see an example of, again, fission yeast, the same cell that I showed in the beginning, where individual filaments are now hitting the cell ends. And as they hit the cell ends, they bend a little bit, and this is a signature of a force being generated, because if you have a stiff filament that you put under force, it will bend. And this force is used by the cell to keep the nucleus, which you don't see in this movie, you saw it in the earlier one, in the middle of the cell. So when the cell grows twice as long, it needs to divide in the middle, and it keeps the nucleus centered simply by pushing off the cell ends, alternatively, in a very mechanical process. So this force-generating capability we can also reconstitute, and this is what you see at the bottom. So there's the sort of grayish movie there is a very old one. When I first started my own group, it's a DIC image that is maybe difficult to see, but there's a single wire, a single microtubule that runs into a microfabricated barrier that we make in the clean room, which provides sort of an artificial cell end or obstacle in the cell. And again, without anything else, we can show that single filaments, when they grow, can generate pushing forces. So you see a more recent version of that in the fluorescence image on top. And simply by using last century physics from the bending profile that these filaments show, we can measure and deduce how much force individual filaments generate. And we can, I'm not showing it, but do the same thing also with shrinking filaments in a slightly more elaborate reconstitution experiment. So this is all still about the biophysical properties, but then we can start to think, knowing what these properties are, how much force they generate, how the dynamics is affected by the forces, we can now also start to look does it help organize space. So we can make different kinds of containers using microfabrication techniques, or in this case, microfluidics, to make spherical containers, cell-like containers with, in this case, just a monolayer, but we can also make vesicles with membrane bilayers, and then show that, again, without anything else, the positioning of these, as they are called microtubule esters, happens spontaneously in the middle of such containers, provided that the forces are distributed in the correct way. So, we reconstitute some of the mechanical processes that you see in real cells with a really small set of components outside the cell. And then eventually, of course, this needs to be controlled. So again, at the top, you see now a movie again in a real cell from my collaborator, Anna Akmanova. She's a cell biologist in Utrecht, originally from Russia. And she's a very big expert in knowing everything about the proteins that actually control these dynamic properties. Because I said that you don't need anything else for filaments to be dynamic, to generate forces. But of course, to make them do that in differentiated ways, depending on what the cell is doing, there is a lot of control. And some of this control is exercised by molecules that specifically travel with the ends of these filaments, because the ends are structurally, chemically a little bit different from the rest. This is recognized as a hub by control proteins. And this is now also something that we can add to our system. So we have the biophysical properties, the confined space, the self-organization properties, and then we are now adding control molecules to see how they can vary the behavior of these self-organized systems, still in a very simple way, and then always comparing to theoretical models of the same system. So this is sort of the story of my life. It's building functional complexity and reconstituting systems with the aim to understand better what is the force balance that allows these systems to organize in functional ways in cells, depending what cells are doing. And so the closest we get to a real spindle so far is just by using, like I said, microfluidic techniques, and many of you are probably familiar with this, or maybe not. It's a very simple way where you can create cell-like containers that just bubbles of water in oil with a monolayer of lipids around it. And then in these droplets, we can enclose the components that we need, of the nucleation centers, the proteins that make these tubes, control molecules, molecules that hook up these filaments to the boundary, the cortex of the cell. So you can really build, by putting the components that you want, minimal systems very easily. And then we can sort of get to a situation if we have two of these enclosed in a container, and we control the forces that are generated in the correct way. It's actually quite easy to get to this typical cell division type machinery organization, which is, to me, remarkable, because we know the real spindle has tons of components that are shown to be essential even to get this organized properly. But if you throw them all out and you manage the forces that are generated properly, you get this organization quite easily back, which to me says that all these other components that are helping there are there, at least in part, to regulate the forces that need to be generated. And of course, what's very different between our system and the real cell, that ours is totally non-responsive. So it organizes in one way, and that's it, depending on how we designed the experiment. And the beauty, of course, about the real thing is that this can change from one type of organization to another, and that the cell knows how to regulate in time and space, that this happens properly. And that's also, I guess, where we want to go, even for this project. So we want to, this is just a static picture. It's set up to have a certain type of organization, but we want it to be dynamic. We want to hook it up to clocks, maybe get temporal control, so that we go through different phases of organization inside these artificial cells by controlling also the machinery that controls this specific organization and manages the forces that are generated at different places in this system. One of the things we're doing right now is to also put in little chromosome mimics. So this is a collaboration with a professor from Dortmund at Max Planck Institute, Andrea Musacchio, where we can take beads and coat them with the components that you normally find on chromosomes that are able to hook up chromosomes to the ends of these filaments. So one of the things we're now doing is understanding how force generation happens at the end of these filaments when they're hooked up to these beads, which are mimics of chromosomes, and then we will eventually ask how do they self-organize within this minimal spindle that we've built. So that's all fine. So that's been the focus of my own research group. There's a lot of collaborations, but still it's sort of a single group, not a single group, but it's a single-minded almost approach to one aspect of what the cell can do, and which taken us a while to get to this point, even with all the collaborations that we have. But that's, of course, only a very little start. So if you think about, if we really want to be serious about building a synthetic cell, this has to massively speed up. And we shouldn't only worry about spatial organization, but we should have a system for energy production, for DNA replication, for protein expression. Well, you can read yourself. So even if we ask the simplest to be possible, which is, of course, not simple at all, to have some artificial membrane container that can grow and divide and transmit information, it has to be able to do all these things with a number of components that we control, and that has to be done in an integrated way. So this is why we're now getting together with different specialists who are each been working on the last many years on reconstituting some aspect of what a cell can do and see if we can integrate our expertise. So that's then the program that I promised to talk about a little bit. So this is the slide that we used to also argue for giving us money to run this program, which took us a while actually. So when we first submitted this proposal, I think it was in 2013, and we were completely slashed down by all the reviewers, said it was just totally naive, impossible, will never happen. That may still be true, but 3 years later we did get it funded, so I guess with more or less the same ideas, although we improved our writing a little bit. So the question, really the big question is, we can say we know the molecular parts, be it in a bacteria or a yeast or a human, we more or less know the code, so in principle we know which players are or how many players should be there, also from the top-down minimal cell. But as I already said before, we really do not have an idea how life emerges from these molecular interactions. Even though we're very good at treating diseases and manipulating cells on the molecular level, it's still very much a trial and error approach because we don't really know how this all works together. And a related question, as I already said, which are the molecular components minimally required, and maybe there's not one answer to that, and it maybe depends on what you ask your system to do. You haven't heard me mention the word evolution yet, but of course a real cell also has the ability to evolve, so that may eventually be something that we need to also consider. For the moment, we're asking, what are the minimal components, or related, what are the minimal number of functions that are minimally required to make something that we might call life, and then you can have a whole separate discussion on how you want to define life. But what we want to do as a consortium is have minimal systems that allow self-fueling or some kind of metabolism, homeostasis, where you make ATP, for example, but you also keep track of osmotic pressure so that it's sustainable, some form of DNA processing, and then growth and division, and again, all based on biological life as we know it here on Earth at this moment in time. So what about our consortium? So what we also propose is sort of a modular approach, which could be really not a good idea, we will find out. But the idea is that we start with these individual functions, the energy, the processing, and the division, and then work on those first, and then see if we can combine them. But it could be that this is maybe not the way to think about it, that we have to think about it in a much more holistic way, and I guess we will find out, but this is how we will start. Also simply because of the knowledge, the expertise, the background that we have, because we have a starting point there as a group of people. So there's biochemists involved. This is a biochemist from the University of Groningen. Who has been to the Netherlands? Everybody? It's okay. Half. This is all the way in the north. We are a small country, so we're all very close to each other. Some would call it one big urban area, but Groningen is as far as you can go from Delft. It's 2 hours by train. And in Groningen, they work on the fueling part, so they have membrane systems where they can have sustained ATP synthesis with a minimal number of components. It's not based on the ATP synthase machine, which is a very complex machine, but a simpler bacterial enzyme network that is inserted in the membrane and that can make ATP and keep it producing ATP for a while. We have people who've been working on in vitro DNA replication, so biophysicists working on the nanoscale machine properties of even eukaryotic replication machinery, so that, for example, the yeast replication machinery is 20-something components that can be reconstituted in vitro, where you can really have DNA copied outside the cell. There's people working on gene expression using, and I bet we will hear maybe more about it later, a cell-free system called the pure system or cell-free extracts. It's still very difficult to get protein expression, so the transition from DNA to RNA to proteins in a cell-free system is still challenging. So it's based for the moment on quite a complex mixture of components that make that possible. But you see here a picture of just a green fluorescent protein expressed in a membrane vesicle by my colleague Christophe Danelon in Delft. And you put a piece of DNA in this membrane, and the machinery will allow you to make proteins that are, in this case, fluorescent. But he also had a recent paper where he was expressing from DNA a molecule which would then copy that same piece of DNA. So the number of components is not completely limited but you can really start to have DNA that by the protein it makes copies of itself. And as I said, we also need temporal control, so making clocks, spatial organization, and there you see my minimal spindle again. Of course, we have to go away from the eukaryotic system to prokaryotic proteins, and we probably need simpler forms of spatial organization than the one that we're working on now. But that's sort of the approach or the expertise that we bring in. And then we also have physicists worrying about the mechanical integrity of the container. So these membranes will need to have some kind of stability if you want them to survive shape changes and division. And then also cytoskeletal machinery, presumably for cell division itself, things that will pinch off these membranes in the middle when cells have to divide. So that's how we start. But of course, there's still tons of open questions. And even if we start by this modular approach, we can ask what's the best design for each of them, and we probably have to, we don't restrict any types of inspiration, so the inspiration can come from bacteria or yeast or anywhere. We can mix and match molecules and strategies. We're not trying to make one particular type of cell, we're making a cell-like system that can do what all cells can do, but it can be based on a mixture of designs. And we can maybe optimize for the rest of our careers one module and then find out that when we start to integrate, it doesn't work. So we have to also optimize according to how things can work together. And this, I have to say, is sort of, I think, the biggest challenge of our project. It's starting to work. We now have a lot of PhD students going between different labs without us knowing, which is the best, I think, and trying to combine these different systems. We have to control nutrient flux. I already mentioned the physical integrity of the container. And then also a very big question, how to coordinate growth replication in time and space. So how do you make sure that even if everything works, that you grow twice as big, duplicate the DNA, and divide only once within a certain cell cycle time? And then maybe the most important question at the bottom is, we will run into tons of problems because we are maybe not going from a holistic approach. So how do we then fix the mismatches and the missing links that I'm sure we will find? So this is the group of people involved. So we will also include a top-down approach. So in parallel with building these things from the bottom up, we will have John van der Oost and Pascal Daran. So they are microbiologists who do engineering of real cells, both yeast and bacteria. And what they will do is to take modules that we've designed that work in vitro and then swap them for the functional ones in real functioning cells and see if it can work with whatever else is there, and then maybe use evolutionary techniques to sort of fix or to optimize or to improve the modules that we've built in vitro. And that will go back and forward with the things that we do outside cells. And then very interestingly and importantly also for this type of project, we have a philosopher, ethicist also involved, and this always sort of gets mentioned all the way at the end of the project, but in this case, even though it's in terms of manpower not a big part of the project, it's mostly very technical, I think it's very important and fun. So whenever new students come to this project, the first thing we do is lock them up with Huub Zwart, and then talk about, should we build synthetic cells? What does it mean? What is life? All these questions that we don't have answers for. But it's in the context of this project also for PhD students a really interesting part. And so, which means that, it's not only about PhD students talking with Huub, there it is, but it is also a project where from the beginning we've been trying at least to find, how do we discuss this project with industry also and society. So we very early on, we had just a small dinner in Delft with some local companies. We have DSM, which is a big synthetic biology company in our neighborhood, and just asked them, what could this do for you in the future, if anything? And that's very useful. So they all think, like we do, that this is far in the future. But if it ever happens, there's lots of things that become possible. And also, on the way, there's a lot of technology that comes out that could be important for them. So that's part of the community building we're also doing. And in the last 2 minutes, so that also is very important for the European project that we're trying to build. So this is now a picture taken last August. again, some people here in the room were there in Delft when we had our first international symposium with the program running. And this was really to think about, how should we get together in Europe and maybe also in the US to make this an even more collaborative project, because we can do this one specific focused thing in the Netherlands, but you can think of many other approaches. You can use chemistry to make molecules that are life-like. If you really want to build containers that have life-like properties to make something, it doesn't need to be based on current biological systems. It could be based on supramolecular chemistry or proteins that are not completely natural or any combination thereof. There's a connection to questions about origins of life, to technology of cell-free systems. So that's what we are try to sort of put together on paper, and we did start that off last year in 2018. We're actually getting together again later this year. We wrote, it's on the next slide, but I will not bore you with it, a little bit of a very naive 10-year science and technology roadmap for the European Union, saying that it's not just basic science, but that it will also give all kinds of possibilities for technological applications. And so we wrote this proposal to ask, okay, can you give us money to organize ourselves? That's the way it works in Europe. If you want to have a big program, you first need to go through a one-year phase where you have to formally be organizing yourself, so not informally, but formally be organizing yourselves. Otherwise, you cannot apply for these big programs. So in the end, we didn't get that formally organizing ourselves opportunity, but we are doing that anyway, of course. So we want to write a serious science and technology roadmap for Europe. Of course, we will not do this alone. And we get together in October later this year to give that shape. And then we will informally go back to the European Union and to say that this could be actually an interesting topic to invest in for the future, in collaboration and also in parallel with similar activities in the US. And even though we didn't get the money to get us formally organized, because of the preparation, it did a lot of good to our informal level of organization, because we got the support from many different European countries, scientists, but also by now from sort of on-paper support for this proposal from 20 companies that I didn't really expect. So this is companies from food industry to fuels to even, of course, from the health industry, but also from pharmaceuticals and even L'Oréal, which is making creams for skin treatment, and who all think that this could indeed have possibilities for the future. So they support us on paper, write very nice letters, for the moment not with funding, but we will see. So that's it. I want to finish with just showing the slide to acknowledge the people that I collaborate with, my group collaborates with on the cytoskeletal projects and the funding sources that have been paying for the research in my own lab. And thank you very much. I'm very happy to be here, and I'm looking forward to a really good day. Thank you.
As you well know, the astronomy community, as part of its mainstream research, is searching for microbial life as part of the mainstream.
Yeah.
But intelligent life is considered outside the mainstream. And my question to you is, based on your experience, do you get the sense that making the first cell on Earth out of a soup of chemicals was indeed a much bigger step than going from the first cell to the more complex forms of life? Because that would argue the other way. That would say, whenever you have primitive life, it's actually much easier later on to make it more complex.
Yeah, so it's a difficult question to answer, so I can only speculate. So I think making, so complex life, intelligent life, at the end is based on cells that are, okay, more complex, and they are bigger, and they have more internal spatial organization, but at the bottom of it, there's of course a lot of that also simple cells can do. So what we are looking for is sort of universal to all life, doesn't matter intelligent or multicellular or single cellular. So, and that's where it starts. So of course, we're not asking how did it arise. We just take what's already there and learn how to put it together in time and space, which is a way easier thing to do, even though still extremely difficult, and we will see if we get there. So this all to say that I don't know the answer to your question, what is more difficult. My sort of intuitive feeling would be that going from once you have a cell to multicellular systems where there's communication and then eventually intelligent life is probably easier, but I don't know. That's just not based on thinking about it too long.
Yeah. Thank you for this very fascinating talk. I have a question. I noticed in your presentation that supercomputing is not so much important in your as a tool to develop, simulate those systems. Are they too far complex, or maybe it's not part of your community to use this kind of tools? I want to understand which is the role of computing in this field.
Yeah, no, you're absolutely right. So I didn't emphasize it, but 3 out of the, so we are a consortium of 17 groups, over 6 institutions, and 6 of them are theoretical groups, and 2 of those do a lot of computational work. But it's different. It's not maybe the supercomputing type of work, and maybe eventually we will need that also. So maybe it's something that we will have to collaborate with people who are more experts in that, because it's in that sense not complete all the expertise that we have. So there's different ways you could think about supercomputing, right? You could also just put all the molecules together in a program and then see what it is able to self-organize. I don't think that makes sense yet. To be a bit more guided in how to do that before you do that, I think would be a good idea. But then eventually, we will need it. There's another big project going on in Delft, which is to build a quantum computer. And so one of my colleagues, Leo Kouwenhoven, he's been pushing for that very successfully, and he always says, okay, once we have a quantum computer, then we can calculate, we can simulate cells and then understand them. And I say, okay, so that we're in a bit of a competition. We try to build them, and they try to build the computer to calculate it. So we'll see. We'll probably need both. Yeah. Yeah.
There is a British architect named Rachel Armstrong who's been looking at an application of this technology that will be of great interest to many of the astronomers and astronautics people here, and that is the possibility of using synthetic life in space, not to build interstellar arcs or big space habitats, but to grow them.
But grow them, yes.
Yeah.
I guess that would be the way to go, yeah. Yeah.
We're going over here.
So you talked a bit about the European and American work and cooperation on this activity. I was wondering, is there a greater world coordination with, say, Chinese and Asian research in this area, or is this just completely separate?
Well, yeah, so not so coordinated, yes, but of course in Asia, especially Japan, there's been an interest since many, many years also in this topic. So yeah, so it's on the to-do list, I suppose. So I think there's a lot of individual connections between scientists and communication, but to get a coordinated, bigger program, not yet. Yeah, although it's certainly something that has been on the agenda in Asia, maybe even longer. Yeah. Yeah.
This is really remarkable. Just a quick question. So you've jumped into the remarkable complexity of the eukaryotes, but what about the prokaryotes and archaea? Are these are simple by comparison, but how well do we even understand the initiation and the process of binary fission?
Yeah, so there's quite a bit of, so I have many colleagues also in this consortium, but also outside who've worked a lot on the mechanical aspects of cell division in bacteria. So there's actually a tubulin homologue, which makes filaments that do a bit different things than the microtubules that we work on, that form a ring at the middle of the cell that helps bacterial cells to divide. So there's a lot known in terms of what is the dynamics of the filaments that are responsible for that, how do they behave, what they look like, but again, not really understood yet what is minimally required to use these polymers to make, for example, vesicles divide, but people are working on that quite a lot. And also our efforts have to switch. We just recently switched from using not the eukaryotic tubulin, but the prokaryotic versions. There's also prokaryotic homologs of tubulin, not the FTSE that I was just talking about that is making the ring, but there's others that do exactly the same kind of growing and shrinking behavior that can help organize space also in bacteria. So if you want to have a cell-free system to express these proteins, for example, we better move away from the eukaryotic versions, which are very difficult to express, and go to the prokaryotic ones. So we're moving towards prokaryotic homologs. And your question was how much is known about the biophysical aspects of that, and quite a bit. I didn't talk about it, it's just because we don't do it in my group, but colleagues are working on that, yeah.
Your approach has been to take the existing structures and reverse engineer them to see if you can then replicate that in your lab. Now, evolution produced these incredible machines.
Yeah.
Can we learn something about how to system engineer by taking some insights from what's happened in nature? Now, we don't have a lot of time, but I'm thinking including evolution, for example, as part of a system engineering step is an interesting one which I don't think is explored.
No, you're absolutely right. So actually in the little bit of top-down approach that we also do in the project, we plan to use evolutionary approaches. But yeah, so it's also, one of the best things about this project is all the discussions we had before writing down the proposal in 2 stages. And sort of going back and forward, what needs to be in there, what is feasible, what we could do with this group. And we sort of left out this evolution part a little bit, but I'm sure eventually we cannot. So that will be, so we explicitly didn't also include this in the initial part, but and that's also, yeah, maybe when thinking about this in a modular way, trying to make these models and putting them together, we maybe at some point cannot get away with just thinking about them as machines, but have to consider, how did they in evolution get to integrate with each other? So yeah, it's still, it's a good point.
In the same spirit as Ron's comment, could you think about recent advances in machine learning and so on as being applied to the problem of making synthetic cells, which in some ways has some similarities to the evolutionary process? So yeah, tell me what Ron said in his head, if you like.
Yeah, so I have to think about a bit more, but that would look sort of in a, use big data of everything that's out there, and then use machine learning to distill some kind of strategies that we maybe not would think about otherwise. So we haven't really invested into thinking into this, but that's definitely, so that's also why there's so many ways that this, that this is a limited approach, but that's what we can do, and which I'm also, one of the reasons I think it's very, very good to have a bigger collaboration across even continents, because all these things have to be taken into account, and we cannot do in one spot everything together, but we somehow have to inform each other by taking all these different approaches. We have time for one more. Okay.
Thank you for this talk. From observing myself, I see not all cells are created equally. A cell in my skin is not the same as a cell in my lung or in my heart. And from YouTube videos, I've learned that this process is of differentiating cells as they grow is called evolutionary development. Mm-hmm. I'm curious, how far away are your current technologies from being able to reproduce that in a laboratory setting?
Far.
And what's the challenge?
No, you're absolutely right. So in a multicellular organism where you have cell-cell communication and tissues, of course, all these cells are different because they read out a different execution program from the DNA. Yeah? But the DNA, at least in all of them, is the same. It's just whatever gets executed from that information is different. And that's in a few levels of complexity above what we're trying to do. So a bacterial cell, even single-cell organisms can live in different states, so they don't also always use the same information from the DNA. And I guess what the minimum we're trying to think is to have something just one-dimensional with just one piece of DNA that codes for the limit the minimal number of components that do the growing and the division and the metabolism. And then comes all these extra complexities to have multiple ways of executing the information to some behavior, and then communication, and then differentiation. So, but then we're much, much further away, and that's why we don't talk about multicellular organisms at all at this point. And maybe just going one remark about the evolutionary aspect. I do have a colleague in Delft who is actually now starting to think about how can you in vitro reconstitute regulation networks that have the ability to evolve, so that have not just a single sequence of events possible, but multiple, where you can try out different sort of usages of that network to be able to every now and then lose a component and still be functional and adapt. So it's a bit of a vague statement, but there are also efforts thinking of how to design simple networks that do have the ability to evolve, and maybe we will need to put that in eventually.